AlphaFold predictions in your work. From a mathematical perspective, AlphaFold is an enormous end-to-end differentiable function with lots of parameters optimized during training process to return the structure of a protein given multiple sequence alignment. From technical point of view, this is deep neural network based on modified transformer architecture. But as a biologist, you don't need to know any details about machine learning. You just need to submit the sequence and get the predicted structure. And what is very important is that AlphaFold also returns confidence metrics, which allows us to judge quality of prediction. I mean, the greatest thing about AlphaFold is that if it's not sure about something, it will tell you this in the form of confidence scores. Yes, AlphaFold is a prediction, but the fact that you can access it right from the start of your research unlocks new possibilities, as we'll see later. AlphaFold was released nearly 3 years ago, open source, with the hope that it would be useful to scientists, anyone working with proteins or genes. We made AlphaFold available in several settings. A call-up interface for generating predictions in the cloud, the cheapest and easiest solution to get taste of AlphaFold. We have open sourced our code, and we produced a large database of protein monomers predictions in collaboration with European Bioinformatics Institute. Currently, the database has predictions for nearly all UniProt sequences, with few exceptions. So today I will reflect on what we have learned during these 3 years of AlphaFold, whether we can rely on the predictions, what exciting applications it has enabled, and what are the keys for successful use, and also what are the limitations of current technology. But first of all, a bit of statistics. About 20,000 of papers citing AlphaFold have been published, and AlphaFold database has received 1.8 million unique visitors. This sounds very impressive, but do we have solid proof that AlphaFold is actually doing a good job? Yes, 3 years later, we have. One of the common questions is whether AlphaFold models are confirmed by the experiment. In crystallography, to solve phase problem by molecular replacement, one needs a search model that is very close to the structure in the target crystal. And you need RMSD less than 1.5 angstrom for at least 30% of the atoms. And therefore, if AlphaFold models could be successfully used as search models for molecular replacement, it proves that predictions closely resemble experimental data. A statistics on that was analyzed by Tom Terwilliger and team. They took more than 200 recent structures from the PDB deposited after AlphaFold release. So AlphaFold never saw those structures. And these structures were solved by experimental phasing, so conventional molecular replacement with structures from the PDB did not work for those cases. And Tom and team tried to solve those structures via AlphaFold models. And as a result, initial molecular replacement solutions have been obtained for like vast majority of cases. I think only 7 cases out of more than 200 have failed. I would say this is very convincing statistics that proves that AlphaFold is doing a great job and predictions are confirmed experimentally. Speaking about cryo-EM, interesting evidence came from the novel validation tool CheckMySequence, aimed at checking structures for potential register shifts. Interestingly, some experimental human-built cryo-EM structures suffered from such errors, but AlphaFold models of these proteins are in much better agreement with the experimental electron density maps, and AlphaFold never saw those maps. So AlphaFold prediction sometimes can be more correct than some human-built experimental structures. As the majority of the PDB structures used for AlphaFold training represent crystal states, it was not obvious how well AlphaFold predictions would agree with NMR data obtained in solution. Apparently, high-confidence AlphaFold structures often fit NMR data as good as, or even better, than original NMR ensembles. For instance, it was found that original NMR ensembles were more accurate than AlphaFold models for only 2% of the cases. And these few models were predicted with low confidence. However, 30% of AlphaFold structures have scores substantially better than original NMR structures. So we believe that these studies prove that AlphaFold is able to generalize beyond crystal state. A method of cross-linking mass spectrometry can deliver structural information in situ. It reveals closely located parts of the proteins in 3D space at proteome scale. In this technique, chemical cross-linkers covalently fix amino acids that are spatially close, and after protease digestion step, resulting short cross-linked peptides are identified and analyzed by mass spectrometry. Combined together, structural constraints obtained via cross-linking provide another experimental validation of the AlphaFold predictions, while predictions allow atomic level interpretation of cross-linking data. So this is very nice synergy and interplay between these two methods. For example, 84% of proteins from human HEK cells had all of their high-confidence cross-linked residues within the non-cross-linker lengths in the AlphaFold predicted structures. Taken together, all these various experimental structural data validate AlphaFold predictions and suggest that they are reliable enough in most cases, and this precision is enough to enable various practical applications. Now I will show you some of these applications. The idea is to illustrate a range of possible AlphaFold uses. Arguably, the highest profile application of AlphaFold has turned out to be in integrative modeling and building huge hybrid structures based on low-resolution cryo-EM and cryo-ET. So if you search through AlphaFold citations in journals like Nature and Science, you find that most of them are using AlphaFold exactly in this way, fitting AlphaFold predictions and relatively small crystal structures into low-resolution cryo-EM and cryo-ET electron density maps for the huge complex. This bridges the gap between low-resolution density and atomic details of the structures and overall accelerates map interpretation. The structure on the left is of nuclear pore complex. A more complete model of the pore was produced by 3 different groups, and we were really excited to see that 2 out of 3 groups used AlphaFold as part of their model building process. Intraflagellar train is another example of the huge structure built with the help of AlphaFold. So conscious of time, this is just very incomplete list of the large hybrid complexes built based on experimental cryo-EM data via fitting of AlphaFold models, as well as crystal structures. Most importantly, community members concluded that better structure reconstruction can be obtained by combining AlphaFold predicted structure models and cryo-EM data. Another exciting application of AlphaFold is studying protein-protein interactions, because ultimately, few, if any, proteins function in isolation. Understanding their interaction networks is a big part of understanding biology. There have been quite a number of similar works done for different organisms. I have picked this one for illustration. So authors run predictions for 65,000 pairs of human proteins and obtained 3,000 high-confidence pairs. Roughly half of them lacked homology to existing experimental structures. Researchers have again combined AlphaFold predictions with cross-linking to validate predictions, and they have found that AlphaFold confidence metrics are extremely useful for quality control. For high confidence predictions, 70% of the interfaces fully satisfied experimental cross-linking data. In another example of more focused work, Lim et al. Were studying the protein donsan, which is necessary for the assembly of CMG helicase in vertebrates. However, exactly how donsan mediated helicase assembly was unclear. So the authors used AlphaFold multimer to screen 70 core DNA replication factors for possible interactions with donsan. And based on the most confidently predicted complexes, they were able to build up a structural model of a preloading complex. And experimental evidence for the model was then obtained from co-immunoprecipitation and site-directed mutagenesis. So these examples illustrate how multimeric structure prediction can shed light on protein-protein interactions, both through large-scale screens and more targeted structure modeling. A big difference between structure prediction and experimental structure determination is that predictions can be generated on a massive scale in a short time for whole proteomes or even for the whole uniprot. AlphaFold database now contains 200 millions of protein structures. An interesting question is whether AlphaFold can predict novel folds and structures that are substantially dissimilar from those available in the PDB. And yes, analysis by CAS, protein classification, and other data mining research in the AlphaFold database, they proved that AlphaFold can produce folds substantially different from those in the PDB. So availability of such database opens up possibility of structure-based search against it, which unlocks a multitude of applications. For example, structural search to identify unknown proteins observed in the experimental data. In this work, cryo-EM density maps of the mycobacterial lipid transporter revealed a previously unknown subunit of the complex in sufficient detail to build polyalanine model of the protein. So like, yes, there is a cryo reconstruction, and clearly there is another protein bound, and researchers had no idea what the protein is this. And they just built polyalanine backbone and performed search against AlphaFold database for proteins with such fold. And they got clear best hit, which is LuCB protein, and this finding was subsequently biochemically validated. So this method of structural search was unlocked by the AlphaFold database, and it was not available before because PDB is quite incomplete. And the last slide to illustrate a wide range of AlphaFold applications. One team used AlphaFold to figure out some details about mechanisms of immunity in honeybees. And this example really made us smile. When you develop technology like AlphaFold, you don't really think about the honeybees. And another example is Baker Lab, which is famous for their protein design efforts. And AlphaFold seems to be useful for prediction and validation of the designed protein structures. So the range of potential AlphaFold applications is enormous. And if you work with proteins or genes, you can ask a question: how can AlphaFold be useful for my project? And you can get very creative. From our perspective, it boils down to one of these two uses. So predicted structures can help you to interpret your experimental data, like fitting into cryo-EM maps. And also, it may help to decide and plan what experiments to do to check a hypothesis coming from the structure. So let us speak about the keys to successful use of AlphaFold. The most important key to success is no doubt use of the confidence metrics. You have probably heard that nothing in biology makes sense except in the light of evolution. So very analogously, nothing in AlphaFold predictions makes sense except in the light of confidence metrics. Please keep this in mind. And if AlphaFold is not certain, it will tell you this. So PLDDT score indicates local confidence around single residue, and predicted aligned error, or PAE, indicates confidence about relative orientation of the larger part of the structures like domains. People usually understand PLDDT score quite well. You contrast blue parts of the structure, and you know that yellowish or reddish parts are either naturally disordered, or AlphaFold is not sure about their structure. Maybe because it doesn't have enough information as MSA or whatever. But look at this example. Both domains are blue and confident, but PAE plot does not show any connection between these domains. It means that domains are positioned relative to each other just randomly. And indeed, they are connected, as you can see, by this flexible linker. And likely, they don't have any fixed position in the cell as well. One domain just randomly floating around another. Maybe it could be stabilized by binding to another protein or whatever. But anyway, PAE plot tells you that these proteins are randomly oriented relative to each other. And it will be very incorrect and misleading to make any conclusions from this particular position of the domains. For multimer predictions, there are additional quality indicators, PTM and IPTM scores, which provide an idea about confidence of the protein-protein interface prediction. You can find detailed information in our multimer preprint. Briefly, values higher than 0.8 represent very confident prediction. Below 0.6, it's almost definitely a failed prediction. And between 0.6 and 0.8, it's a gray zone where prediction can be correct or wrong. And please note a very important exception. Some proteins have huge disordered segments, and such segments impacts and dramatically reduces overall PTM and APTM scores. So you can have very correct and very valid interaction between two small domains, but overall IPTM score can be very low due to massive disorder around these folded domains. In that case, please pay attention to the PAE plot. If PAE suggests that two domains from two proteins are confidently positioned relative to each other and make good connection on the PAE plot, this is the proof of the predicted interaction. So don't be confused by low PTM or IPTM score if you have a massive disorder. Another key to successful use, in large part, the widespread adoption of AlphaFold has come thanks to the work of tool developers who have integrated the AlphaFold into standard software used by the structural biology community. Chimera X, a model building software for Aquarium, supports importing of the AlphaFold models, and it displays nice dynamic PAE plot, and you can even run AlphaFold predictions from Chimera X. Result is also supporting AlphaFold models, as well as many other software suites. So one of the keys to successful use is using available tools to make your life easier. We also have been really surprised by how quickly community adopted AlphaFold and built on it for new applications. That's been incredibly exciting for our team to see, and that's an absolute win. For instance, AlphaFill that came from PDB Redo developers, it complements AlphaFold predictions with ligands were available. And also AlphaFold predictions were integrated into specialized databases like those of membrane proteins and glycosylation. And new software adopts AlphaFold for such uses as in silico computational pull-down screenings, like protein-wide screenings for protein interactions. Also, please note CollabFold, alternative collab interface to AlphaFold. It has more options available than our very basic collab. So using the right tools is another key to unlock full AlphaFold potential. So a typical workflow looks like this. First of all, you may want to check predicted structures for the monomer of your protein in the AlphaFold database. You will also have an idea whether AlphaFold predicts reliable structure or not for your case. Just look at the quality indicators. And you may also learn about disordered segments and domain arrangement of your protein. Then you may use CollabFold to model oligomeric form of your protein if it forms oligomers, or you can model protein-protein complex or try to model alternative conformations using MSA tricks or templates. And if you plan to run massive predictions like interaction screenings for hundreds of structures, then you may consider installing AlphaFold locally in your powerful workstation with a state-of-the-art GPU. Please note that local installation might be challenging. AlphaFold runs only on Linux. It requires 3 terabytes of disk space for the databases, plus it requires modern GPUs, a lot of memory, as memory is the limiting factor for the size of the complexes AlphaFold can model. The good news is that there are alternatives to buying and managing expensive hardware. You can simply provision AlphaFold on a virtual machine in the cloud. All the major cloud providers are compatible with AlphaFold and provide detailed instructions for AlphaFold installation on their VMs. It's very important to note that AlphaFold inference workflows can be very compute intensive, but there are ways of managing this. Google Cloud has developed a reference architecture leveraging Vertex AI. This makes the end-to-end process as simple and as cost-effective as possible. Up until now, AlphaFold may seem like some miraculous solution to numerous biological problems, but it's not some magic, of course, no. It's a technology, and as any technology, it has inherent limitations. So let us speak about AlphaFold limits. AlphaFold 2 can't fold nucleic acids or DNA or RNA complexes. This was just out of scope of AlphaFold 2. It was trained specifically for proteins, polypeptides. Also, AlphaFold 2 was not designed to predict multiple conformations for the same protein, but it appeared that through MSA tweaks, you can do this. Also, the performance is not guaranteed. AlphaFold 2 often fails to predict effects of point mutations, and it was not designed to predict ligand and ion binding and post-translational modifications. Also, AlphaFold 2 is not aware of the membrane plane for the transmembrane domains, and as a result, membrane and non-membrane domains often have random orientations, which is indicated by PAE plot. AlphaFold 2 rarely predicts correct antigen-antibody interactions, unfortunately, due to their nature. And also, AlphaFold 2 has reduced performance on orphan proteins, for which no or very few homologs are available to build multiple sequence alignment. So, our plan for the future developments is to lift at least some of these limitations. So, this list actually works as a plan for our future work, and we are exploring possibilities of tackling these questions. The latest version of AlphaFold is aimed at modeling of the whole variety of the molecules present in the PDB. It can model proteins, DNA, RNA, small molecule ligands, and ions, and also post-translational modifications of the proteins and chemical modifications of the nucleic acids. Latest AlphaFold can natively model multi-component complexes that involve all these different molecule types. Speaking about ligands, the model outperforms standard docking methods. For nucleic acids, it outperforms automated methods, but for RNA modeling with human expert, RNA modeling done by human experts still performs a little bit better. And please note improved predictions on antigen-antibody interactions. These predictions require running many seeds, so for better sampling of the left-hand space. Oops, what was said? Sorry. So, I'm sorry.
Leave call button. Double tap to activate.
Oh my God. So, these are a few examples from the test set. AlphaFold never saw these or similarly enough structures before, and nevertheless predicted these structures quite well, including positions of the small molecules and nucleic acids. It's just examples for the illustration. One may say this is a cherry picking, but I would say that we are quite strict with our validation protocols. So these examples are quite fair demonstration of what the latest AlphaFold version is capable of. I need to admit that not all the limitations of the original AlphaFold were lifted up. We are still limited in our ability to predict affinities of the interactions and the effects of point mutations. We are also dependent on human expertise and functional experiments to connect predictions to functional understanding and understanding at the levels of cells and the whole organism. And that's quite a major gap. And I would like to conclude my talk with an important statement. In no way do we see AlphaFold as a rival or a substitute to the experimental methods. We believe that initial hypotheses provided by AlphaFold may facilitate and guide experimental structure determination, complement low-resolution data, and overall accelerate structural studies. Also, hypotheses based on the AlphaFold prediction may help to advance new research, including biochemical and cell biology experiments. That's our hope. And if you don't have much experience with AlphaFold yet, we have recently prepared practical introductory course in collaboration with AmblEBI to facilitate the learning. So please try it. It's available on the AmblEBI training website, or you can just Google something like AlphaFold course. So it only remains to thank all the people who made AlphaFold possible, the team at Google DeepMind, our wonderful collaborators at AmblEBI, and of course, the wide community of AlphaFold users. and all the authors of the above-mentioned research and developers integrating and building upon AlphaFold. And thank you for your attention.